Good morning. My name is Adam Boxer, and I'm honored to have been asked to present to you today about tau therapeutics for neurological disorders. Here are my disclosures. So what I want to tell you about today is the current status of tau-directed therapies for different neurological diseases. And the questions that I would like to address are, why should we be developing therapies to target the tau protein? Who are the patients most likely to respond to treatment? What are the therapeutic approaches, and when will a tau therapy be most effective? And finally, how do we determine which tau therapy should advance to pivotal clinical trials? And really, I also want to tell you a bit about recent advances in tau biomarkers for Alzheimer's disease. And this is really an advantage for developing tau therapies in Alzheimer's disease. And with these new biomarkers, they are enabling us to take clinical trial designs that have previously been used in oncology and apply them to neurodegenerative disease. So most of us, I think, became aware of tau protein in the setting of Alzheimer's disease. And this is the classic description of Dr. Alzheimer's patient Auguste D, who had a severe early onset form of Alzheimer's disease, possibly caused by a presenilin mutation. And when he studied her at autopsy, her brain at autopsy, he found that many of the neurons, or where the neurons used to be, were filled with these tangles that he called neurofibrillary tangles. And these were subsequently found to be made up of mostly of tau protein. And so many of us think of Alzheimer's disease as the archetypical, or maybe the first described tauopathy, but that's not actually true. It turns out that we might say a lay neurologist, Mr. Charles Dickens, long before Dr. Alzheimer's, described a patient with a different tauopathy, progressive supranuclear palsy, in one of his novels. And so his really perfect description of PSP Richardson syndrome was a chilled, slow, earthy, fixed man, a cadaverous man of measured speech, a man who seemed as unable to wink as if his eyelids had been nailed to his forehead, a man whose eyes, two spots of fire, had no more motion than if they had been connected with the back of his skull by screws driven through them and riveted and bolted outside among his gray hair. And so Charles Dickens really got this idea of the supranuclear gaze palsy, even though he didn't have a medical way to describe it, but he got it perfect. And so many people have been thinking about and have noticed tauopathies really for hundreds of years. So why is there so much excitement of developing therapies for tau-directed therapies for different neurological diseases? Well, in Alzheimer's disease, but also in PSP and other diseases like chronic traumatic encephalopathy, we know that the amount of tau protein that is deposited or measurable at autopsy, both the amount and the anatomical location are strongly correlated with the clinical features during life. So the more tau protein you have, the more severe the clinical symptoms, and also where it is located anatomically seems to correlate very strongly with the types of symptoms or the clinical phenotype that we observe. Excitingly now, with new molecularly targeted PET ligands, one is called fluortaucipir, we can now detect the accumulation of these same insoluble tau aggregates in living patients. And so in the spectrum of Alzheimer's disease, you can see that when people start to develop symptoms of Alzheimer's disease using this tau PET ligand fluortaucipir, you start to be able to measure increasing amounts of insoluble tau in their brain. And when they have full-blown dementia from Alzheimer's disease, there's really quite a bit of this tau protein. So again, similar to what we see with the Braak stages of Alzheimer's disease that I showed in the last slide, we now have biomarkers of insoluble tau protein that are very useful, particularly in Alzheimer's disease, for clinical development and for staging disease. So I think many of us also thought about tau protein as a target for therapeutics based on the work of Leonard Mucke and co-workers. And this is an early study where in a Alzheimer's transgenic mouse carrying a human amyloid precursor protein mutation, you can see that about 15% of these mice die after about 6 months. But if you genetically knock down the levels of the tau protein, you see that the mice, many more of the mice survive. Not only that, but in other mice with genetically reduced levels of endogenous tau protein, they become resistant to the onset of seizures from the neurotoxin kainate, requiring higher doses to induce a seizure related to the amount of tau in their brains. So now we just about 15 years later, there are now human experiments going on. And this is a phase 1 clinical trial with an antisense oligonucleotide where it's been shown that it is safe and well tolerated in patients with Alzheimer's disease to knock down genetically the amount of tau protein with this antisense oligonucleotide to about the same level that you would expect by a heterozygous transgenic mouse knockout. Now, we don't yet know whether this has had the same sort of clinical benefit that you see in a mouse, but so far, after months of treatment, we know that this is safe and well tolerated, and we're all eagerly awaiting the clinical efficacy results. So another reason why tau is an excellent therapeutic target relates to genetics. And so to remind you, the tau protein is alternatively spliced. This is a representation of the DNA and gives rise to 6 alternatively spliced protein isoforms that contain either 3 microtubule binding domains, so 3R tau, or 4 microtubule binding domains, 4R tau. And in a normal brain or in someone with Alzheimer's disease, you have an equal ratio of these 3R to 4R isoforms. But in certain neurological diseases, for instance, corticobasal degeneration or progressive supranuclear palsy, we have a over representation or a selective accumulation of 4-repeat tau. Not only that, but there are rare autosomal dominant families, and you just heard about one from Dr. Beauvais earlier, where there are mutations, point mutations in the tau gene that lead to severe early onset dementia called FTDP17. Interestingly, depending on the type of mutation and where it's located in the tau gene itself, you get different phenotypes. So with these intron 10 mutations, you actually you often get a Parkinsonian phenotype that looks like CBD or PSP. And interestingly, conversely, when you look at sporadic PSP or CBD with genome-wide association studies, you find that the strongest genetic link in these sporadic tauopathies is to the tau gene itself, particularly a regulatory region around it called the H1 haplotype. And one particular subregion of this H1 haplotype, H1C, is thought to lead to increased expression of the tau gene itself at the mRNA level. These different tau mutations now have been used to develop transgenic mouse models and other non-clinical models that have been used for identifying new therapies that are now in human clinical trials for different neurological diseases. So what's another example of how the heterogeneity of different tau mutations relates to a clinical disease? Well, you heard from Dr. Beauvais about this N279K family that he's been following, but other families followed by Dr. Beauvais and coworkers at the Mayo Clinic have mutations that lead selectively to a 3R, 4R tau that is similar to what you would find in Alzheimer's disease. Interestingly, even though these patients do have no amyloid in their brain, they have a clinical syndrome that looks like Alzheimer's disease and actually very strongly bind the tau PET ligand for taucipir, similar to what you would see in Alzheimer's disease. Interestingly, when we compare the rate of progression of this form of these types of tau mutations with other tau mutation carriers, they also have a slower rate of progression, very comparable again to what you would see in Alzheimer's disease, but not like a frontotemporal dementia, which progresses much more quickly. And so based on all of these types of data, there is a current model for tauopathies and how the tau plays a role in neurodegeneration. And we think that really that there is a toxic gain of tau function that occurs. This leads to neuronal or glial dysfunction and death. And this toxic gain of function may involve misfolding of the tau protein. And the one current hypothesis is that this misfolded tau may spread from one cell to another, and this might explain the spread of tau through the brain and the progression of symptoms that we see in different tauopathies. We also know that there are endogenous mechanisms within cells that can block the accumulation of these toxic forms of tau. And we designate the different tauopathies based on the type of proteins that we find in autopsy. And in tauopathies where the only pathogenic protein that's detectable at autopsy is tau, we call primary tauopathies. So examples would be tau mutation carriers or PSP or CBD. Secondary tauopathies are like our Alzheimer's disease, and we call them secondary because we think the tau toxic gain of function is occurring secondary to the accumulation of another toxic protein, and in this case, for Alzheimer's disease, beta amyloid. And then we also think that there are that tau may play a contributory role even in neurological disorders where you don't find tau protein deposited at autopsy. So for a number of different synucleinopathies, we know from genetic studies that the tau gene is linked to disease in synucleinopathies, even though you don't find tau protein in autopsy. And this suggests that for all of these different types of disorders, potentially reducing toxic tau gain of function might be a viable therapeutic approach. So when we zoom in now at the cellular mechanisms of tau toxicity, to summarize a lot of work by a lot of people, we think that tau is transcribed from the MAPT gene, as I told you earlier. This produces a soluble form of tau protein. There are post-translational modifications that occur that may lead to the formation of toxic forms of tau. These may be small aggregates, and the aggregates that may be toxic in and of themselves may be cleared by endogenous mechanisms such as autophagy or other forms of proteostasis. But if not, the aggregates or these toxic forms of tau may be spread to other cells and may template the formation of further tau aggregates in those other cells. Again, if those aren't cleared by the next cell, they may lead to toxicity and cell death, and eventually the formation of these large aggregates that we measure at autopsy or by PET. So one of the real questions here is how much time does this take, and which portion of these cellular processes are occurring in the brain at different times during different diseases, and this is really an unknown at this point, I would say. So what types of therapies are entering the clinic or in clinical trials right now? Well, there are genetically targeted therapies that can block the production of tau protein and by blocking translation, so antisense oligonucleotides or gene therapy against the tau gene itself. There are a variety of different small molecule enzyme inhibitors, such as kinase inhibitors or glycanase inhibitors that may block toxic post-translational modifications. There are small molecule aggregation blockers that may prevent the formation of these small toxic aggregates of tau. And there are new molecules that are coming into the clinic that may enhance the clearance of toxic forms of tau. One type of these molecules called a proteolysis targeted chimera or PROTAC. But the most advanced form of therapies currently in clinical trials are immunotherapies, and these are either active vaccines to tau protein or anti-tau monoclonal antibodies. So if you have one of these exciting new anti-tau targeted therapies, what disease would be most efficient to develop the therapy, or in which diseases should we be thinking about doing clinical trials? And I would suggest to you that there are really three main indications that we're considering right now, and we can compare these based on their genetic relationship to tau, the efficiency of doing a clinical trial, the availability of pharmacodynamic biomarkers of tau protein itself and the prevalence of the disease. And so the largest number of clinical trials have been done in Alzheimer's disease, even though it's not genetically related to tau, and it takes large numbers of patients and a long period of time to do a clinical trial. It's really the biomarkers that we have that are leading us to really pursue many clinical trials in Alzheimer's disease. This is not to the exclusion of other tauopathies such as PSP, and there are good reasons to do clinical trials in PSP, particularly the genetic relationships, the fact that we can do faster and smaller clinical trials for clinical efficacy in PSP, but this is rare and we don't have good biomarkers. Ideally, we'd like to do clinical trials in tau mutation carriers because in effect, they are like human tau transgenic mice, but they are very rare and very heterogeneous. And so it's going to be hard to do clinical trials in these individuals, although, as I'll tell you in a minute, we're working on methods to do this. And so really, as I just told you, right now, if we think about the cycle of clinical development of therapeutics and preclinical development, Alzheimer's disease really has the inside track, and that's because of likely the availability of these tau biomarkers that can inform not just clinical trials, but the preclinical efforts to refine the development of new therapeutics. So why is it so hard to think about doing clinical trials in tau mutation carriers? Well, this is some work from the FTD Prevention Initiative, which is currently a collaboration of an autosomal dominant FTD network called Genetic FTD Initiative in Europe, and the All-FTD study in North America. And what you can see, for instance, if we look throughout the world for all known tau mutation carriers here in purple, that in the Americas, really, we were only able to find roughly 300 tau mutation carriers. And so this isn't much when you consider many of them might not actually be at the right stage to be in a clinical trial. But we haven't been too discouraged, and in other rare diseases, we've been encouraged by the FDA to think about using innovative methods to really take the most efficient way to follow disease progression. And one way is to generate what are called Bayesian repeated measures disease progression models. And here we can take participants or individuals with different tau mutations or other FTD causing mutations and do a clinical trial that involves a spectrum of different individuals by creating a disease progression model that we can use to interrogate the effects of a new therapeutic agent. So how does this work? Well, for this work that I'm going to tell you about, we took data from the GenFree network using individuals who either were asymptomatic mutation carriers, CD clinical dementia rating scales of 0, or with mild to moderate symptoms of these different mutations, including tau mutations, or from all FTD. And we looked at different clinical rating scales, including the clinical dementia rating scale, neuropsychological tests, a general marker of neurodegeneration called plasma neurofilament, as well as different volumetric MRI measures of brain atrophy. And we compared this large number of different autosomal dominant mutation carriers with their non-mutation carrier family members as controls. So what do these models look like? What do the data look like when we combine across this global, these two global networks? Well, at first, what we wanted to do is make sure that the disease progression looks similar in European FTD mutation carriers and North American FTD mutation carriers. And here what you can see is that in blue, we have the All-FTD network. In green, we have a GenFree network. And this is, again, a global rating scale called the CDR-NACC-FTLD sum of boxes. And really, the two lines are really superimposable. And we designate 0 in these disease progression models as the earliest detectable clinical symptoms of disease by a clinician. And here in orange, you see the non-mutation carrier control family members. Now, we can also look, when we combine across both networks, the different disease progression by the different types of mutations. And here you see that the tau mutation carriers and the C9 open reading frame mutation carriers actually have a less fulminant or form of disease because their CDR sum of boxes scores rise less quickly than in the progranulin mutation carriers. We can also look at a different clinical rating scale, a neuropsychological test of executive function called TRAILS-B. And again, you see that as compared to progranulin mutation carriers, tau mutation carriers have an attenuated onset of executive function deficits, and C9 mutation carriers are sort of somewhere in the middle. And these are actual raw data from individual patients that are superimposed on the model. When we look at biomarkers, this is we can look at volumetric MRI changes. And here, these are very important for the models because here again is the onset of clinical symptoms. And you can see that we're starting to see brain atrophy 10 or 20 years prior to the onset of symptoms. But again, the rate of progression of brain atrophy differs by the type of FTD causing mutation. And again, with frontal lobe volume changes, tau mutation carriers actually have slower atrophy than progranulin mutation carriers. Also, tau mutation carriers have slower accumulation of plasma neurofilament values. And in progranulin mutation carriers and C9 mutation carriers, you see that even 20 years before the onset of symptoms here, that we can start to detect this neurodegeneration in the blood. So now if we summarize all of these different types of endpoints in the disease progression models, we can start to identify how we would put together a clinical trial, for example, our tau mutation carriers. And here we might be able to include both pre-symptomatic people before disease age onset of, or the onset of disease or disease age 0 by using brain atrophy measures, because we can reliably detect atrophy in the medial temporal lobe up to 10 years prior to onset. Unfortunately, unlike in progranulin mutation carriers or C9 mutation carriers, we cannot use neurofilament light chain for pre-symptomatic disease, although it starts to increase. And the nice thing is that we can compare using these models the effect sizes of the different clinical scales at different stages of the disease. And so we're using this disease progression model to plan a tau mutation carrier trial of new tau therapies. So I want to switch now to PSP, and just to illustrate that for clinical trials in PSP, we have a big advantage when we want to look for clinical effects of a tau therapy. And this is an example from the Davunitide clinical trial. And here we were very well powered, 90% power to detect a 37.5% treatment effect of this drug with a 1-year trial with about 150 participants per arm. Unfortunately, the drug had no effect, but if we look, for example, at similarly mild to moderate Alzheimer's disease as compared to PSP, we need a much larger number of participants on the order of 600 per arm as opposed to 150 per arm, and we need to follow them for a year and a half as opposed to 1 year to have comparable power. So that's an advantage potentially of testing tau therapies in PSP as opposed to Alzheimer's disease. We can do it much more efficiently. But really, I think where the advantage comes in for Alzheimer's disease is with biomarkers. And so as I showed you earlier, we can detect these aggregated forms of tau in the brain parenchyma, either at autopsy or with tau PET. But increasingly, we're learning that there are soluble forms of tau protein that are released from these processes. And these are measurable either in the cerebral spinal fluid, these, by and large, are mostly N-terminal fragments of the tau protein, because most of the tau protein is cleaved in the brain parenchyma, and the C-terminal regions form the aggregates. But we can detect different forms of these, either N-terminal or central tau fragments in CSF. And excitingly now, using blood tests, and particularly these phosphorylated fragments of tau protein are very specific and sensitive for Alzheimer's disease pathology. So how does this relate to Alzheimer's disease, and how do we know that tau plays a role in Alzheimer's disease? Well, a nice example are in mutation carriers of the presenilin gene, and there's a large cohort, I think as many people have heard about, in the country, Colombia. And here, you may have seen this article in the New York Times a few years ago. Most of the people in this presenilin 1 large family, where there are thousands of members who are being followed, get sick with symptoms of Alzheimer's disease in their 40s. But there was one individual who lived well into her 70s who actually was a presenilin mutation carrier, but died from cancer. And this is a picture of her, and these are her daughters. And this individual was very generous and donated her brain for autopsy, but also did a lot of work in this research study. And so you can see that this is her brain, and this is an amyloid PET result. So she had a lot of amyloid in her brain when she was older. But even though she had a lot of amyloid, which we think causes Alzheimer's disease, she had very little tau protein. And so this was her in her 70s, whereas the average family member who was in their 40s when they get sick would have a lot of tau protein. And so there was something that allowed her to resist this Alzheimer's disease causing mutation. And this turned out to be a protective mutation in the apolipoprotein E3 allele called the Christchurch mutation. And so here, even though she had a known Alzheimer's disease causing mutation, she was able to prevent symptoms. And this was strongly predicted by this tau biomarker, tau PET. So as I mentioned, now really there's a lot of excitement that we can measure tau not just by PET or by CSF tests, but also in the blood. And so here's an example of some work that we did using samples from UCSF and from the All-FTD network. And here you can see that in a variety of different neurodegenerative disease syndromes, either Alzheimer's disease, spectrum disorders, or FTD, that individuals with Alzheimer's disease, and these green dots indicate that they're also amyloid PET positive, tend to have highly elevated, up to 4 to 5 times higher than control, levels of phosphorylated tau 217 in their blood, whereas we don't see this in other forms of dementia like frontotemporal dementia. Not only does this work in clinically to diagnose patients, but also at autopsy, where you can really use this phosphorylated tau in blood to find people who have neuropathologically confirmed Alzheimer's disease. And looking at receiver operating curve statistics, this is a highly accurate way to differentiate Alzheimer's disease from frontotemporal dementia. I think really the most exciting potential is that eventually it may be possible to do these blood tests very inexpensively and potentially in remote or underserved populations. And so a p-tau test may only cost, on the order of hundreds of dollars when it becomes clinically available, whereas a tau PET scan that I showed you was currently cost $5,000 to $10,000. And these rendered images show that the amount of plasma p-tau is strongly correlated with regional tau PET uptake, with R values up to 0.9 here in yellow. And so it may be that for certain types of research and diagnostic purposes with these new plasma biomarkers for Alzheimer's disease, we won't even need to use PET scans anymore. But really what I wanted to tell you about today is that we can use these biomarkers for clinical development of tau therapeutics. And this is a hypothetical pathway for developing a new drug in humans. And I won't go through all of this with you, but we can think about early phases of development, where when we think about a new tau drug, where we're initially interested first in whether a drug is safe and well tolerated. But really, when we get to sort of intermediate stages, the key question is, does a drug have a pharmacodynamic effect? And for the tau therapies, what we want to know is, does a new tau therapy actually affect tau protein levels in living humans? And I showed you one example with ASOs at the very beginning of the talk, but we've been able to use other tau biomarkers to look for the effects of tau therapies in other clinical trials. And so I want to give you some more examples of this and really how it's related to what we know about tau therapeutics. So we were very excited a few years ago to be able to look at the first of the monoclonal antibodies to tau, and this was gosuranemab, which was an N-terminal tau antibody. And here in PSP patients, we could show that we could use a competition binding assay to look at CSF levels of N-terminal tau fragments and show that even with very low doses delivered peripherally to the PSP patients, we could bind almost all of the N-terminal tau fragments with gosuranemab as compared to placebo-treated individuals who had all of their N-terminal tau was unbound. Interestingly, however, if we looked at other fragments of tau protein, these were not affected by the antibody. And so this may have foreshadowed the fact that when these came to clinical trials in PSP to look for clinical efficacy with gosuranemab, we found absolutely no effect. And so here it was a very well-powered clinical trial that was completed a few years ago showing that even though we fully engaged tau protein in the CSF in these patients, there was no clinical benefit over 52 weeks of treatment. And interestingly, similar antibodies, tilavanamab and semrinimab, had no effect in another PSP trial and in multiple trials in very early Alzheimer's disease. And so really we're left with the question of why, because all of these antibodies had very prominent effects in either transgenic mouse or in preclinical cell culture models where we could show, or where individuals could show that the antibodies blocked this tau spreading that I mentioned earlier, which is thought to be integral to the progression of tauopathies. And so where we are now with tau therapeutic development is we're asking, well, why didn't these initial tau therapeutic approaches work? Did we have the right target? Did we test them in the right tauopathy? Maybe PSP or Alzheimer's disease was not the right one. Did we get to people too late in the stage of disease? Did we get the right dose? Did enough of it get into the brain? Or maybe we need to think about other therapeutic approaches. Well, one thing that all of these antibodies had in common, and as well as a fourth antibody that was just reported to have failed in an Alzheimer's clinical trial, is that they targeted the N-terminus of the tau protein, whereas newer immunotherapeutic approaches to tau are targeting the microtubule binding domains. And this may be important and may explain why these initial attempts were not effective in having any sort of clinical benefit in tauopathies, based on recent beautiful work using cryo-electron microscopy. This is from the group of Cher Shores and co-workers. And here you can see again a schematic of the tau protein, and they've been able to take these fibrils of tau protein and develop three-dimensional models. And what you see is that really the key part of the tau protein that accumulates and forms the core of these insoluble tau fragments that accumulate autopsy are at in the microtubule binding domains and at the C-terminus. And interestingly, it turns out that there are different structures of these insoluble tau deposits in different tauopathies. So Alzheimer's disease is different from corticobasal degeneration, which is different from PSP. And so the initial tau therapies, tau therapeutic monoclonal antibodies that failed, all targeted this area that really was unstructured and not specifically associated with disease, but the newer monoclonal antibodies that are now entering clinical trials may be more successful because they're associated with this disease-associated area of tau. But it also raises the question, because the structure of this core is different in different tauopathies, will these newer antibodies work equally well for each tauopathy, or will we need to test them in different tauopathies to find the right indication? And so to think about that, we can start to use new clinical trial designs taken from oncology. And so one idea is a basket clinical trial where we use where we could include multiple tauopathies and look at, for instance, one of these new tau therapies in different tauopathies to see which one might actually respond the best. And so we've done this with a microtubule stabilizing agent and shown that you can include Alzheimer's disease patients, corticobasal degeneration patients, and PSP patients in the same clinical trial and look for differential effects of an agent. And I won't show you the results of the trial, but just to say we found very different effects. They're all negative of this tau therapy, but that we could define these different populations using biomarkers and include them in clinical trials. And I think increasingly we're going to see this for different tau therapies. I mentioned that we now have plasma tau biomarkers, and really they're mainly only useful for Alzheimer's disease, but in certain frontotemporal dementia or frontotemporal lobar degeneration syndromes, they may also be useful because they can rule out Alzheimer's disease as a cause of frontotemporal lobar degeneration. And this is particularly a problem in corticobasal syndrome, where up to 40% of patients may actually have disease that's not caused by pure tauopathy, but is caused by Alzheimer's disease. And here we use plasma p-tau values in our NIH-funded 4-repeat tauopathy neuroimaging initiative to identify individuals who likely had underlying Alzheimer's disease as a cause of their corticobasal syndrome. And we looked longitudinally with volumetric MRI at how these two different populations of corticobasal syndrome patients, those who likely have true corticobasal degeneration versus those who have a mimic that's caused by Alzheimer's disease, what you can see is that the patterns of brain atrophy over time, over the course of a year, are very different in the true tauopathy patients caused by CBS FTLD, or probably they likely have underlying CBD or PSP pathology. They have much more prominent volume loss in the prefrontal white matter and in the brainstem, as you would expect, as opposed to patients who have Alzheimer's disease, where you see these changes posteriorly in the parietal lobes and the subcortical white matter. And so this may allow us to do much more efficient clinical trials of tau therapy, specifically for corticobasal degeneration tau pathology. But I mentioned that we are learning from autopsy studies that even though drugs like gosuranemab didn't have any clinical effect, they were getting into the brain and having very subtle effects that were measurable by neuropathology. And so because we can do a very efficient clinical trial in PSP, many of these patients were followed for years with in our phase 1b study in an open label extension, and some came to autopsy. And here it was possible to demonstrate that actually tau accumulated in astrocytes, that the antibody actually was binding to tau and getting into astrocytes and being taken up into vesicles within the astrocytes. And this led to a inflammatory reaction that was visualizable as an atypical reactive gliosis in microglia in these cells. And so we know that despite the fact that gosuranemab was not an effective therapy for PSP or Alzheimer's disease, it seems to be getting into the brain and doing something, but maybe it's just not clearing enough tau to have an effect. But there's starting to be some mysterious findings coming in from clinical trials, and these data are hot off the press with two of the antibodies that failed in early stage clinical trials or in mild prodromal to mild Alzheimer's disease. And here in more advanced Alzheimer's patients, there was an effect of one of the N-terminal antibodies particularly on memory, and it may have slowed changes on memory on the ADAS-Cog in these patients, although it had no overall clinical benefit. Whereas another N-terminal antibody actually may have caused individuals to get worse. So this was gosuranemab, that same antibody I just told you, in prodromal to mild Alzheimer's disease. And so there are these very subtle hints that N-terminal antibodies, even though overall they're not efficacious for either PSP or Alzheimer's disease, may be having a biological effect. And as we tease these out more, we may be able to learn more about the biology of tauopathies in humans. And one idea is that these antibodies actually, that neutralizing tau in the extracellular space may also have extracellular and extraphysiological effects. And so it turns out that gosuranemab and some of the other antibodies, zagotenemab, actually in cell culture models block the effects of tau fragments on leading to depolarization and increased firing rates of tau protein on cells. And it may be that in addition to causing intracellular neurodegenerative effects, that tau protein in the extracellular space may affect the excitability and network processes. And so this is work from, again, from Leonard Mucke, showing that if you knock down tau, that you can show that it changes the neural network properties in different cell culture models. So still a lot to learn, and I think that the clinical trials of these tau therapies, even though they have yet to demonstrate any form of efficacy, may be starting to teach us about the biologically relevant mechanisms of tau protein in human disease. I think the most exciting thing that I really want to leave you with now is that in Alzheimer's disease now, we are starting to see that two of the tau biomarkers are actually seem to be strongly associated with clinical benefits of therapies. Unfortunately, these therapies are not tau therapies, they are anti-amyloid therapies. And so this is from the donanemab clinical trial. Donanemab is an anti-amyloid therapy. And here they saw in the primary endpoint a clinical composite called the IADRS, that there was a clinical benefit in slowing disease progression as compared to placebo. And this was strongly correlated with a decrease in the accumulation of tau PET signal or insoluble tau in the frontal lobes of these patients, and a decrease, a really prominent decrease in plasma p-tau levels. And so we think now that at least for Alzheimer's disease, we can now potentially use these tau biomarkers, either tau PET or plasma p-tau, in future clinical trials, not just of anti-amyloid agents, but of tau therapies. And so the NIH-funded Alzheimer's Clinical Trial Consortium is planning a new trial. It's currently not funded yet, but under review based on this idea. And this is work from my collaborator, Keith Johnson, showing that really around the onset of symptoms of Alzheimer's disease, you have a rapid accumulation of this insoluble tau accumulation in the brain. And so the rate of accumulation changes from a very indolent, slow accumulation of tau to a very rapid uptick in this tau protein, here designated by amount of tau accumulation per year. And Keith and Risa Sperling call this the catastrophe. And so in Alzheimer's disease, because we have this great biomarker tau PET, we can find people right before the onset of this rapid explosion of tau pathology in the brain measured by tau PET, called the catastrophe, and start to think about intervening with a tau therapy. And so in this planned Alzheimer's disease tau platform trial, hopefully that will be funded next year if we're lucky, if our study section members find this trial compelling, we'll be looking at the effects of either of two different tau therapies that hasn't officially been chosen yet, alone or in combination with an anti-amyloid therapy, and looking for the ability to block this catastrophe by tau PET or measured by plasma p-tau. And so I think this may be a different way that we can efficiently identify whether any of these tau therapies are going to be effective for neurological disease. So we're very excited about this. And again, a reason why Alzheimer's disease is sort of pulling ahead in the race for developing tau therapies. So with that, I think I told you a lot about tau therapies and a lot about tau biomarkers, but what I want to really emphasize, since I'm a clinical trialist, is the only way to find effective treatments is through doing these clinical trials. And that to really understand tau protein, even though we know that it's strongly implicated in neurological diseases, we need to do these clinical trials. So most of the new tau therapies that may find a role in many different neurological diseases work by reducing tau gain of function. And that increasingly we have tau biomarkers that are useful for diagnosis and for clinical trials. particularly the new blood tests may be transformative, but these really mainly only work for Alzheimer's disease, and we don't have good tau biomarkers for PSP or for other tauopathies yet. So far, none of the tau therapies have been successful for any disease, but we're starting to understand what a successful tau therapy might look like in Alzheimer's disease based on successes recently from anti-amyloid therapy trials. And we also know that from natural experiments, like the patient I showed you with a protective ApoE mutation, that if we block the accumulation of tau protein, we can see less of an accumulation of tau by PET or plasma biomarkers, and this may translate or strongly correlate with better clinical status. And that's sort of the strategy that we're taking in upcoming Alzheimer's disease tau clinical trials. So with that, I just want to remind you that even though we haven't had any successes yet with tau therapies, it's still very early days, and we still have a lot to learn, both about the therapies, but also about the biology of tau protein and neurological disease. So with that, I want to thank all my wonderful colleagues at UCSF and Adam Staffroni, who did the disease progression modeling, Lise Tijan, Renaud Lajoie, and Julio Rojas, who did a lot of the plasma biomarker work, and support from the NIH for all this work, as well as the Tau Consortium, and my colleagues in the ACTC who have developed this new Tau platform design with me, and particularly Keith Johnson. And with that, I think we hopefully still have some time for questions. Thank you.